Hello and welcome to Fascinated by Fungi Live. Today I'm going to be speaking with Richard Tehan, who is an avid mycologist. He's actually an academic mycologist. He's also an incredible naturalist, a musician, a dad, a graduate student, does all sorts of stuff. I was lucky enough to meet him up in Oregon a couple of years ago at a little gathering of mushroom friends, and I was very, very lucky to see him speak last year at Soma Camp. And he does some really interesting work on the secondary metabolites of fungi, particularly with some cordyceps-like fungi, which is something that I think a lot of people have interest in, including myself. So I'm going to be diving hopefully fairly deep, but also keeping it, accessible and interesting and entertaining. And I'm really excited to talk to Richard. So let me see if I can get him on the call here. Per usual, I think the way we're going to do this is we'll probably be kind of chatting back and forth. If you guys have questions you want us to kind of spin in, we'll do our best to address them. But generally, it's going to be Richard and I kind of going back and forth. So there he is. Hello, how you doing?
Ahoy, I'm pretty good. How are you, Gordon?
I'm excellent. Oh man, I forgot to grab a beer. Darn it. I got a little LaCroix. It's not quite the same, but
all right. Well, cheers.
Yeah. Cheers, man. I'm glad. Oh, is that one of my stickers on a glass, huh?
It is.
Nice. Very nice. So yeah, I was just telling people that you are an accomplished everything. It seems like you have many things that are impressive, but you are one of the sort of few academic mycologists that I've gotten a chance to meet, which is really interesting.
Well, I'm not sure I would call myself a mycologist. I think I'm more a chemist with mycologist envy.
Okay, that's good. That's a good thing to remind yourself of, because I think a lot of people look at mushroom hunting and say, oh, this is inscrutable. You have to have a degree to do this. And almost every single person I meet and talk to that's into mushrooms has no official background in this stuff. And so that's, even for someone who's doing mycological research, you don't consider yourself a mycologist per se. You're just a chemist who's taken an interest in the fungi and has kind of transformed that into a research project and stuff. So
yeah, and there's tons of people like that call themselves an ecologist, but they study the ecology of fungi, and that's just how things go. And I think that might be due in large part to the fact that we don't have mycology programs. We have biology programs and ecology programs and things like that. And
right. And I think that's a darn shame, because I get a lot of people saying, hey, I dream of being a mycologist. I'm like, well, cool, but it's going to be a circuitous path to get there. It's not quite the same as you saying, well, I want to do organic chemistry. I'm like, okay, great. We got a whole pathway for you to go down and do organic chemistry for, as long as you can handle it, kind of thing. But, so I guess speaking of pathways, I'm curious, what did bring you to graduate school? Do you have sort of myco motivation that led you there? Or was it an interest in chemistry, or like, how did you end up working on the stuff that you work on?
Well, let me just let you be forewarned. My internet isn't working too great here, so okay. I think you were breaking up a little bit, but I think I got the gist of it. And the way I got to be working on fungal chemistry is, let's see, do you want the long version or the short version?
Whatever you feel like sharing, it's all good.
Yeah, I was originally a chemistry student, just interested in nature. So I was studying chemistry. I really like chemistry and figuring things out. It's a lot of logic puzzles, and structural elucidation is really cool. But I'd always liked nature and the outdoors, and hiking, and actually fishing was my obsession prior to mushrooms. And one exquisitely rainy year, it was so rainy, and I was living in upstate New York at the time. We had so much rain that it was like a 100-year high, and we had federal disaster relief from all the flooding. But I went out hiking one 4th of July to go actually do some fishing, and had a little digital camera to take pictures of a fish that I caught. But I ended up training the camera on all the mushrooms that were ubiquitous because of all that rain. And it just got hooked. I hiked out the woods the next day and bought my first field guide. And then I started reading about mushrooms. And I'm like you, I am fascinated by fungi now, because once you start diving into it, it's just endless novelty and interesting things. And I found myself as a chemist just really well positioned to understand one of the more interesting things about fungi, which is that they make a lot of really cool molecules. They make drugs, they make medicines, they make psychedelics, they make poisons and toxins and all sorts of really cool things. So I just kind of married my two interests, and now I'm studying the chemistry of fungi.
Yeah, that's awesome. I mean, again, I think it's always interesting to hear people sort of intro into the world of it. And it's, sometimes it is you have a family friend or a thing, but sometimes it is just being in nature and saying, what is this thing? And it is incredible how I think so many humans are drawn to the fungal fascination, just because they are sort of that weirdly ephemeral alien looking thing that has weird colors and textures. And it looks, when you run into a mushroom and it really captures your imagination, it completely shifts your worldview. They're like, this is real. This isn't just a piece of plastic that somebody put out here to mess with me.
Nobody told me this existed. That's my reaction to a lot of fungi that I meet.
Definitely.
Exactly.
So I've been trying to think about how to summarize your work, because I did see you talk at SOMA, and it's interesting. But you also gave 2 talks at SOMA that were geared into sort of slightly different directions. Some of it was you look very intently at particular elements of secondary chemistry, and then other parts of what you do is sort of larger metabolomic screening type stuff, where you're just trying to identify a whole suite of molecules. Can you talk a little bit about what your particular project entails, and can you give me the elevator speech for your PhD research here?
Yeah. So I guess I kind of do some chemistry research and some biology research, and the chemistry is looking at secondary metabolites made by fungi. And my thesis research is on one particular group, on genus Tolycopladoium, and that's a group of fungi better known by their synonyms Cordyceps and Cordyceps, are more familiar names. The current name is Tolycopladoium, and this is a really interesting group of fungi that have evolved to have various ecologies. Sometimes they're a little versatile, where they can have more than one ecology, where they live in the soil, and then they live on the insect. And they probably all live in the soil to some degree. You can find any fungus in the soil. Mm-hmm. But then some of them are known as mycoparasites, and some of them are known as endophytes. And we have these different lineages with specifically they have different nutritional modes. So they're getting their energy from plants or from other fungi or from insects. And my research was focused on seeing how did secondary metabolism evolve within this group, and how did it evolve, particularly along ecological lines. So are there certain secondary metabolites?
Common ancestor that you were trying to say, okay, we're going to go from here and look at how these metabolic pathways have shifted slightly, or enzymes have been modified, kind of thing?
Yeah, and it's kind of rare that we can identify the entire metabolic pathway. We're taking it, one molecule at a time, and the goal is to try to find, to try to link secondary metabolites to their biosynthetic gene clusters. And sometimes we can say, okay, here's this metabolite, it's made by this set of genes. And then we can say, okay, that gene is found in all these fungi, or homologs of it, and so forth. And then, yeah, so it's discovery of new molecules. It's linking molecules to ecologies, and a little bit of chemical ecology. So seeing maybe what those chemicals are doing in nature, and also it's drug discovery. So doing biological characterization of these molecules and seeing how they might be therapeutic, if they can kill cancer or bacteria. So there's a lot of aspects to it.
Yeah, I think that's why when I saw your talks, I was just blown away. But I was like, oh my gosh, there's so many interesting things that you could, seemed like each aspect of your PhD project could be its own PhD project. And that, I was sort of surprised that you're not at the point of already running your own lab, because holy cow, there's so many questions to be asked. And I think I dealt with this in my PhD, that by the time I was done, I had more questions than I'd started with, which, from a student perspective, you're like, oh crap, I failed. But from a scientific perspective, or even maybe potentially from your PI's perspective, you're like, that was a very successful PhD if you got to the end of it and you ended up with more questions than you started with, because you learned enough that you were able to start asking more questions and kind of going down different pathways as you're describing. So.
Oh yeah, yeah. That's definitely true for me. I'm kind of behind on writing up a lot of the things I've already done, and I need to do that. And I have some exciting papers in the pipeline, but then I just have tons of preliminary data that will be inherited by the next generation of graduate students. And maybe I'll take some with me wherever I end up going, and continuing down these lines of research.
Yeah. I was going to ask, so you're probably drawing somewhat close to the end, and or you're starting to feel the little bit of fire on your backside when they're like, "Hey, you're still here," and you're like, "Yep, I'm still here."
Oh yeah.
That had to do with a lot of my favorite professors from my core classes. That they saw me in my 6th or 7th year, and they're just like, "Ah, Gordon, huh? You getting out of here?" And I was like, "Yeah."
I'm feeling a little old for this.
Yeah.
Yeah.
But yeah, do you want to stay in academia? You think about doing a postdoc? Are you going to wheel off and start your own lab and do secondary metabolite screening on metabolomes of fungi? What's your dream?
Yes. All the above. Yep, that's what I want to do. I just want to find myself wherever I can keep doing the research I want to do, which is precisely this, and as well as some other stuff too. I'm really interested in the taxonomy of this group of fungi as well, which I kind of started doing when I started studying their chemistry. And I got focused on, well, if I pride myself as a mushroom hunter. If I'm gonna know something about the chemistry, I better know a whole lot about the classification of these fungi, and then that all just exploded into another line of research too, because actually, as it happens, the North American Tolycopladoium are not precisely classified yet. So
right. And so this is gonna trip me up. Tolycopladoium, yeah, something like you're saying it perfectly. I wrote this word up and I was trying to figure out how to pronounce it phonetically, and I was like, I'm just gonna let you say it over and over again. Anyhow, these are really cool fungi because they come from, as far as I understand, a cordyceps lineage. So they're cordyceps-like fungi, but they made the jump from, usually cordyceps are very specific to their insect hosts, and these made the jump to actually eating mushrooms or truffles in particular. And this is interesting to me because anyone who's familiar with the game Last of Us is predicated on the idea that cordyceps made the jump to humans. And as a way of sort of trying to calm people down whenever I post mushroom videos, people are like, "Oh my God, it's going to be the next Last of Us." I'm like, don't worry, cordyceps are pretty specific to their insect hosts. They usually don't range outside of the insect. And here I am, eating my own words, eating truffles, as I'm seeing cordyceps jump from, an insect species, not to just another insect species, but to an entirely different form of life, which is eating truffles. So can you speak a little bit to that biology? And you mentioned ecology too, of them potentially eating other things and being based on their nutritional source. How did that happen?
Oh yeah. There's a lot to say about that. So let me dive into a whole lot of their ecology here. So you have the Cordyceps sensulato. This is Cordyceps in the broad sense, and that's a lot of fungi. That's there's something like 1,500 described species within this, and those are classified within 3 different families. So not all of those are in genus Cordyceps sensu stricto, but they are in various genera that have been described, and it's not one monophyletic lineage actually. So there's fungi within there that have lost their Cordyceps essentialness. So they've evolved and they've diversified and they've jumped to new hosts. So the Elaphocordyceps or Tolycopladoium, those are ones that have really expanded and in their truffle parasitism. So there's 18 described truffle parasitic Tolycopladoium species.
Wow.
And not all Tolycopladoium species are parasites of truffles. Some of them are parasites of insects. So it's thought that the ancestral ecology of the whole genus was insect pathogenic, but that's actually not very well resolved. We need to do more sampling of that. But the hypothesis goes, and it's called the host habitat hypothesis, is that these insect parasitic Tolycopladoium species, ones occurring on cicadae actually, they would find themselves on a cicada nymph, which is associated with the roots of a tree. It's right there. The cicada are sucking the sap out of the roots, and they're there and they're full of sugar and they're getting eaten by these fungi, and the fungi are, shooting a mushroom out of their head. But also right next door in the same vicinity is maybe a truffle. This really fatty, actually, they have a lot of fat in them, in the peridium.
Nice.
And they're full of nutrition, and they're right there, and they have a chitinous exoskeleton or peridium, just like an insect, and it's another tender morsel lying in wait. So the hypothesis goes that these insect parasites jumped to the other kingdom, which you're just asking for it by being there. And I wouldn't speak too soon. Gordon, in saying that cordyceps can't jump to humans, because there is an entire genus within the Cordyceps sensulato that is primarily known from humans. So there's a cordyceps, a human cordyceps. It's called Perengiodantium, and I think it was actually just this year was the second species in that group, second species in that genus that was described, Perengiodantium americanum. And there was one called Perengiodantium album. And both of those have had multiple clinical isolates, usually like skin infections. One woman died from the infection of this. She had a brain abscess where this fungus was isolated from, and they deduced that this fungus killed her. So, and it's not the first cordyceps to kill a person. There's another one called Purpuriacillium, which is distant from Perengiodantium. It's actually close to Tolycopladoium. It's in the family Ophiocordycipitaceae, and that's another one that is considered an emerging human pathogen. And there's been dozens, maybe hundreds of clinical isolates now. And it usually also causes skin infections, sometimes infections of the eye, usually after an eye surgery. Mm-hmm. And there's been several other infections of just deep tissue infections and a vaginal infection of an outwardly immunocompetent host. So, usually fungal infections people are immunocompromised. Well, this is one that infected someone that had no signs of immunocompromisation. So, cordyceps grow on people.
Okay, well, thank you for terrifying me and everyone watching. That's good to know. In terms of the infection, do you think there's anything, is it coming from us digging around to the soil, or do you think it's just like it's in the environment, it's potentially in the air, it's spores that just land on skin and are opportunistic, or how do they make the decision to infect?
Good question. A lot of them just have got to be opportunistic, because you can also get infections by some of these generalists like Bovaria and Metarhizium that can kind of occur on lots of insect hosts and often just found in the soil. And a lot of them have been spread around because they've been used as biocontrol agents. So we use them in agriculture, and they're used for controlling pests. And that may or may not be how some of these human infections have occurred by being spread. We're also using a whole lot of antifungals in the environment, so in agriculture. So now we get these environmental fungi that are resistant to our antifungals, and we don't have a whole lot of a huge arsenal of antifungals. We have the azoles and a few others, but we use those in agriculture. So we're breeding fungicide resistant.
Yeah, it's the same thing as what we're doing with bacteria, but we have even fewer tools for fungi than we do for bacteria, because there's far fewer fungicides than there are antibacterials.
Yeah, so that's why you might see an increase in some of these case reports. Also, we're just better mycologists these days. Well, we're more equipped mycologists, I should say, and we can identify these fungi better now and classify them better. And now we have these case reports and data accruing. So, maybe Purpuriacillium infections happened all the time, and we're only just now noticing.
One thing to point out, though, is it's still going to be like a little scratchy red dot. Or I guess it can be terminal, but generally you're not going to be turning into a zombie and having a cordyceps thing come out of your head. Not yet.
Yeah, I'm not aware of any forming a fruiting body on a person. I think there is a case of a Schizophilum commune, the split gill polypore, actually fruiting on a person.
Oh, really? Were they made out of cellulose? I
think they had extremely high blood sugar.
Oh, okay.
And it's like in the palate of their mouth, something like that. Yeah.
Interesting. I've heard of it happened to some poor guy who took a really hardcore dose of chemo and had his system wiped from antibiotics and other stuff like that, and he went out and had a beer like you're drinking. And Saccharomyces cerevisiae actually got established in his gut microbiome.
Wow.
So then anytime he would consume sugar, he'd end up kind of wasted, which sounds fun up until you realize that eating anything makes you drunk, which not fun.
Wow.
So, there's examples of stuff like that. And I think there was a cool Radiolab podcast recently. I don't know if you listened to it, but they were talking about this increase in human fungal pathogens as a result of the fact that the global temperatures are rising a little bit. And that humans having a fairly high body temperature is one of the things that makes it harder for fungi to colonize us. And so the fact that the environmental temperatures are rising means that these fungi are being selected for things that can survive at higher temperatures.
Oh, cool.
Then it means it can, and it's also potentially more humid and wet, and we get sweaty, and then you get fungal disease. So, eek.
Well, I hadn't heard that, but yeah, that makes sense. Interesting.
So I guess I'm curious, because I love cordyceps are just one of those. I think you can capture anybody's imagination with a cordyceps, because it's a little
spore that lives in the soil. It sees an insect and it starts forming conidia, and it gets inside and it starts growing mycelium through it, and it turns these insects into little, zombies that they're marching along. And the craziest thing to me was learning that the locomotion of these infected insects was actually due to the mycelium, almost like hydraulically moving them along, as opposed to just, it's not manipulating the insect's brain, because the insect's brain is still totally intact. In fact, it's almost isolated from the rest of its body. But the mycelium is the one that's like clomping along and choosing how high up on the branch to grow, to walk up before it shoots the little thing out of its head. So I'm curious, if that's what happens to insects, what do these Tolycopladoium do to truffles? And what does that infection process look like? And what kind of chemistry happens in that?
Well, yeah, so those are some of the things, some subset of those things I'm hoping to try to find out.
To figure out. You don't have to have all the answers. I'm just...
Yeah, a lot of it is just not known. I don't think Tolycopladoium are manipulating the behavior of truffles. Truffles don't have behavior, but
That's true.
and behavior manipulation is only found in a few lineages, actually a few independent lineages of Cordyceps. So especially in genus OSU Cordyceps, you see the zombie ant fungus, species complex, and really all that is not well established. There's some evidence to show that, well, it doesn't the mycelium doesn't colonize the brain, that there is mycelium development around muscles, and some of it actually penetrates muscle cells. There is secretions of these extracellular vesicles, possibly containing secondary metabolites. It's not known. There's some really cool SEM images of all of this, but there's a lack of chemical analysis. So I think that's a job for a fungal secondary metabolite chemist, I think, to investigate further. So yeah, a lot of really interesting questions.
Maybe
how these fungi
should throw some money at that and
totally.
and get somebody to do this research.
Absolutely, I concur.
So I guess I am curious, because you said it doesn't modify the behavior of a truffle, and I'm gonna put this out because this just popped in my head. Fungi infecting other fungi generally cause metabolic changes. So I'm thinking specifically of like a lobster mushroom. So we have like a base species of Lactarius or Russula that gets infected by hypomyces, and there's a paper out there that describes some of the genetic and the metabolic changes that are happening in the first couple centimeters of tissue that seem to change upon infection. So in that sense, the mold is modifying the mushroom's behavior by like parasitically castrating it, but it is changing the texture and the metabolites and the flavor, and that could be expressed as maybe mushroom behavior. And truffles are well known for their stink, for their complex set of aromatic secondary chemistry that helps them attract an animal to help them spread their spores. So I'm curious, with these infected truffles, do they still get dug up? Are they still as aromatic? What does that look like?
I don't know the answer to that, but the group of truffles that they parasitize, it's specifically one genus. They're not infecting really the truffles we think of as gourmet truffles.
Okay, so
they might be gourmet to a squirrel.
Have to worry about. Yeah. Okay.
No, they're not gourmet to a squirrel. So they infect truffles in genus Elaphomyces, and to us, Elaphomyces are not fragrant. They wouldn't taste good, I don't imagine. They're really tough, and the gleba's just powdery, chalky. I don't think you would get anything out of it. But presumably, a squirrel loves it because squirrels eat them. I don't know if there's volatiles that the squirrels can detect, and I don't know if they would eat a Tolycopladoium infected truffle. So, sorry, I don't know.
Yeah, that's okay. This is, this stuff that I end up sitting out like late at night thinking about this, and normally I don't have someone that I can actually get a coherent answer from. My brain's just kind of turning over on itself about this sort of stuff. But
I should say some of the Tolycopladoium, which is an interesting observation, some of them will make the truffle abort, and you'll never find mature spores in those truffles that are infected by this Tolycopladoium, and other ones won't.
Yeah, it's like parasitically castrating the same kind of way.
Yeah. And that actually is one of the identifying characters of some of the species. If the truffle's it's always aborted in one species, not in another.
Very cool. So we have, the other thing that kind of blew my mind is the adage that I'd heard and sort of researched about was we don't have a lot of cordyceps diversity here in the United States. The only one that's really well known and somewhat characterized is Cordyceps militaris. That's mostly an East Coast thing. And again, I had my mind blown by talking to William Padilla Brown, Mycosymbio, where he was like, I've actually seen Cordyceps milletularis on like 50 different insect species. And I was like, holy cow, whoops. I've been going along saying that, they don't infect that many kinds of insects. And here's one that's clearly diversified. So it's no longer a specialist as much. It's become a generalist on lots of insects. And then you blew my brain even more when you're like, oh, there's plenty of West Coast Cordyceps. We just don't know what they are, and you're out there characterizing some of them. Can you talk about a few of the things you found? Have you logged any collections? Have you, is this stuff getting published? Are people aware of what you're finding?
Oh yeah. There's I would never say that North America doesn't have Cordyceps diversity. It has tremendous diversity. It's a hotspot, in fact.
Okay.
But I would say Southeast United States is a Cordyceps hotspot. And a lot of Cordyceps have been described from Eastern United States. Not too many have been described or recorded at all from the Western United States. Some from the Pacific Northwest, but not really many. And at this point, I would shout out my lab mate Connor, who has single-handedly found just tons. He's found a lot. He's a really good finder of Cordyceps, among other mushrooms, but myself too is, and when I put in the hours and the effort, Connor and I both go out a lot together and find a lot of Cordyceps actually. In fact, got some right here if you want to see.
Oh heck yeah!
I had them kind of incubating in a humid chamber, just some damp paper towels because they were kind of immature. And if I just leave them in here for a while, they will mature. But the other day I was at Mary's Peak near Corvalis, Oregon, and can't really see it too well, but this is a spider, and I'll take a better picture with my camera.
Is that one that's on your profile? I think I posted a spider that had a bunch of sort of white fuzz on it. Is that the same thing?
Yes,
yeah,
that would be Gibellula species. And actually, I have another species altogether, and both of these are on the undersides of leaves. And this is something I'm working on, and I think it's in the Canthomyces. But
cool.
It's tiny. So some Cordyceps are huge, some Cordyceps are tiny, and but most of them are tiny, actually.
And that's a good lesson, because I think a lot of people might go out and they'll look around and they're like, I don't see any ants with giant orange things sticking out of their heads. You're like, yeah, but there might be something here. Maybe it's on the underside of a leaf, and you wouldn't even notice it unless you get up in there with a macro lens kind of thing. So. Oh
yeah, you have to look, and you have to look in the right habitats, and you have to look very carefully and closely. I use a flashlight often in dark, damp places. Mm-hmm. Connor the other day crawled into this log, that just had like a tiny hole, and just crawled halfway into there and like found some really, really cool Cordyceps. He shared them on iNaturalist. But I should mention I started an iNaturalist project to catalog the Cordyceps that are occurring in North America. And we have nominally 47 species of Cordyceps sensulato in United States, Canada, and Mexico. And there's a lot more. There's a lot of species that have been described and never observed, and just kind of described and forgotten. We've never seen color photographs of them or any photographs. There's also a lot of undescribed species, and I think some of these will constitute that. And I'm studying that, yeah. And I should show you. I have one of my personal herbaria here, just full of collections. It's my Ziploc bags with dried specimens. Most of those are Cordyceps. A lot of them are ones Connor and I collected, and some other ones that other people sent. Damon Talley, who just sent several spider pathogenic ones that I want to look at. But I got my microscope at home since COVID, and I've been hard at work on that because I have reduced access to the lab. I needed something to do at home, but looking at them under a microscope and sequencing and all that is forthcoming, and definitely there are some descriptions in the pipeline.
Very cool. Yeah, I mean, I guess that's what I'm so excited about, is that there's sort of this idea that there's not a diversity, and there is. It's everywhere. It's just hard to see, and it's hard to notice, and it's kind of it's the same idea as you're walking along the woods and you miss something unless you're like on the level of the ground, or you're on the level of the trees, or like, I did scuba, and it was amazing. If you swim through the same area at slightly different depths, you'll see completely different life and different things interacting with you. And it is just a matter of opening your eyes, paying attention, And then importantly for science, documenting, right? It's collecting, it's documenting, it's doing the work to figure out type specimens and look at spores and all that kind of stuff. And the ultimate, I mean, not the ultimate goal, because in some ways you just want to catalog biodiversity, but the power of that biodiversity is that within one of those species that you just cataloged and you put it into a secondary metabolite screen, boom, maybe we have a new antifungal, maybe we have a new antibiotic, maybe we have a new cancer drug. I mean, there's this incredible world of secondary chemistry that fungi do. And I listened to an interview with Tom Volk. I think you heard it too. It was on Welcome to Mushroom Hour. He's a great podcast. Go check him out. But he did a great interview with Tom Volk where they talked about secondary metabolites as sort of being waste products, which I hadn't really thought of. And I'd spent a lot of time thinking about the secondary metabolites of yeast because it turns out to be some of the main constituents of the aroma components in wine. And so I'm very used to thinking about yeast secondary metabolites and how that relates to organoleptic properties of the wine that you drink. And in that sense, I was aware of them being a waste product, but in a yeast, it's a single-celled thing in a liquid suspension, so that just volatilizes out and gets, sort of integrated into the wine. But hyphae and mycelium are living in soil, and they're producing their waste products. And I think Tom said something really interesting. He's like, why would you spit out a waste product that you're just going to sit on? You don't shit where you eat, right? So they end up putting a lot of those secondary metabolites, all those waste products, into the fruiting body. And that's why you see this disparity between what's in the mycelium versus what's in the fruiting body. And so I thought that was just sort of a fascinating idea that these secondary metabolites are waste that's getting shuttled elsewhere. And I assume it's sort of the same thing with a cordyceps. Can you talk some about, I mean, there's the insane diversity of secondary metabolites, but can you talk a little bit about that production of metabolites and specifically why it's so interesting in cordyceps and cordyceps-like fungi?
Well, yeah, so genus Tolycopladoium in particular is an interesting system just because it has that, that evolution of ecology. So if you want to ask the question, how does secondary metabolism relate to ecology? How are they using these chemicals in nature? It makes a really good system because we can look at these. There are genomes sequenced for the various nutritional, fungi with the various nutritional modes, and we can see how the genes differ. And I'm the chemist on the project, and this is a collaborative thing with biologists who are looking at the genomes. I can see how the chemistry differs. And let's see. I'm sorry. What was your exact question? I kind of forgot the question there.
I guess I was asking specifically what makes the secondary chemistry of cordyceps so sort of intensely interesting. Why do they make these bioactive compounds that we don't necessarily see in other mushrooms?
Yeah. Okay. So as I was getting at, the ecologies have evolved, and they're kind of closely related fungi, and we can see how closely related fungi change their chemistry, and really it's a good system to compare within. But also, one important thing there is that we know that they have a lot of secondary metabolite biosynthetic genes. And that's something true of fungi in general, is fungi are prolific producers of secondary metabolites. And now that we have genome sequencing and more genomes of fungi are being sequenced and uploaded, we can scan the genomes of those fungi and do genome mining and look for the genetic signatures of secondary metabolism, look for the particular genes, secondary metabolite biosynthetic genes. They look a particular way to an algorithm. And we can predict what they're going to make. And importantly, when we predict what Tolycopladoium can make and other cordyceps sensulata, we predict that they make a lot of secondary metabolites, a lot more than we know. So they have this genetic potential. They have all these genes that don't have secondary metabolites characterized. So part of my job is to go and find out what else can they make. And one reason for that is that a lot of these genes aren't always expressed in laboratory conditions. Right. So we got to try to poke them and prod them and manipulate them in various ways to try to get them to express those so-called cryptic gene clusters, the ones that aren't turned on in the lab. And there's ways to do that. We vary the media conditions and growth, and people use epigenetic modifiers and things like that. So Tolycopladoium also is famous as the source of an already important drug. So if you've ever known anyone that has had an organ transplant or tissue transplant, they might have taken cyclosporin, which is an immunosuppressant that was discovered from Tolycopladoium, and that is used to prevent the rejection of organs. It was actually discovered from Tolycopladoium inflatum, which is the fungus originally isolated from soil. And at the time, they didn't know that this fungus had a Cordyceps teleomorph, as they call it, the sexual stage. They didn't know this fungus's life cycle included an insect pathogenic time. And so we were using this drug. It was an FDA-approved drug, clinically used, before we even knew that the drug was made by a fungus to overcome an insect. So that's one reason we want to maybe really focus on ecology and say, well, what else can we know about what ecology can predict about the secondary metabolite potential of a group of fungi? So our entomopathogens are a particularly rich source. Our endophytes, actually it seems endophytes are a phenomenal source of novel secondary metabolites. And
that I guess, again, from ecological perspective, that makes sense, right? If your job as an organism is to take over another organism and or live inside of another organism, you are going to be having to respond to a lot of chemical cues and relating again to that system, whether you're living antagonistically to it or living sort of in symbiosis with it or somewhere in between, which it seems like kind of the relationship between plants and fungi is always this weird push and pull, and there's no real true symbiosis and there's no real true parasitism. There's always, maybe there is true parasitism. Our malaria are pretty gnarly, but there's always
Perengiodantium.
There's always some form of sort of like pushing and pulling a little bit. And so it does make sense that, cordyceps and things that are sort of attacking insects trying to take over that system are going to have stuff that might inhibit RNA synthesis or inhibit, immunosuppression. There's going to be things that the molecules that they're using and that they've evolved over time to help them do what they do. And it's not that the evolution was driven necessarily with intention. It was more driven out of necessity because this is how they run their metabolism. It's how they run their life cycle. And every time the plant or the host would develop some sort of defense, then the fungi could come up with some enzymatic chemistry solution to kind of overcome said defense. And that's how we've ended up with this, arms race over time of amazing secondary metabolites that fungi are producing, and not just as waste products, but as like intentional things to help them do what they need to do. So that's fascinating to me. It's
totally.
Cool.
And they have to have an evolutionary advantage because they require a lot of energy to make. So they require a lot of resources from the primary metabolite pool and energy. So it takes a lot of energy to biosynthesize these molecules. It takes a lot of ATP. Mm-hmm. So if these weren't advantageous to the fungi, they would just be wasting a lot of energy, and these would get selected out. But they're clearly selected for, and they certainly serve some important function in a lot of cases. In a lot of cases, we just don't know what that function is. That's a job for a chemical ecologist, I guess, to work out. Yeah. And that's one of the aspects of Tolycopladoium I want to work out, is what they do with these secondary metabolites, and taking it one molecule at a time. But we know they make many dozens of secondary metabolites. So in Tolycopladoium, they have anywhere from 30 to almost 60 predicted secondary metabolite biosynthetic gene clusters in their genome. And we know, from some of these, a handful, maybe 5 to 10 at most from a single species. There's a lot of discrepancy between what is known and what is predicted.
Right. Well, and like you said, getting them to express these things in a lab can be troublesome because, just like you said, they don't want to expend excess energy. That's part of their biology. It's like, if I don't need to do this, why would I do this? And you see that a lot with fungi and bacteria and other things. They're like, if I don't have to deal with this kind of stress, I'm going to choose not to. I might even kick out the gene cluster in my genome that helps me deal with this thing if it hasn't shown up in a couple generations and it's no longer a concern for me. Get rid of the genes and then keep chugging because I've now pared down and made myself that much meaner, leaner, and faster kind of thing. So getting these secondary gene clusters in the lab to express and being able to study them sounds like that's no wonder why you're doing a PhD. It takes time. It's complicated.
And actually,
go ahead.
Well, in some cases, the expression is the problem where one molecule is overexpressed, but that's not what we're interested in, and I can't separate it from the other molecules I want to. So actually, someone published a characterization of a biosynthetic gene cluster, a functional characterization, where they knocked out each gene at a time and found what each gene did. And I saw that one of the knockouts results in the lack of production of this important compound, this well-known compound, and that's the compound that's a problem for me. And I asked the researcher for that strain, so they sent me this knockout strain. So I didn't have to do any knockouts or genetic manipulation of these fungi, which is kind of tricky and very time-consuming. So they just sent me the strain, and now that's what I'm working with. I work with that. So I'm working on isolating the rest of the chemicals besides that one.
There was just like there was some huge peak that was difficult to elute the different things. You were able to take out like the main signals so you can see closer to the noise, kind of small stuff.
Exactly, and get more yield of the other secondary metabolites.
Very cool. Is there a secondary metabolite that you've discovered that you would guess has an interesting bioactivity, or is there a family of things? Is there something that's really caught your interest and you're excited to tell people about?
Sure, yeah. So one of the groups I've really honed in on are called peptide biotics or peptide balls, and that's broken down as it's spelled PEPTAIB. IOTICS. So the AIB, the AIB is an acronym for a particular amino acid, alpha amino isobutyric acid, and it's this alpha dialkylated amino acid. So it's just two methyl groups sticking out of the alpha carbon. If you know your amino acids, you have the amine and the acid, and then the side chain. Well, the side chain is just two methyls, and that's really uncommon in amino acids. And these fungi incorporate a lot of uncommon or non-proteinogenic amino acids, right? And so these are ones that aren't in the proteins. They're only in these exotic molecules, like peptide biotics. So it's a peptide. My headphones might be dying. Can you hear me still?
I can hear you fine. You froze for a second, but you're good.
Okay, yes.
So they're incorporating pieces of peptides with small molecule.
Yes, so they make peptides, and it's a peptide that has AIB and they're antibiotic. At least a lot of them have been found to have antibiotic activity, killing bacteria and fungi. And I did isolate some new peptide biotics or peptide balls. Sometimes they're called because they have an alcohol N-terminus or C-terminus rather. And so those form alpha helices, and they kind of form like a corkscrew that affects membranes, and they make membranes leaky.
Membrane is like a bunch of phospholipids all moving around like this, and if something interdigitates and disrupts that membrane, suddenly you have a pore that's allowing stuff to diffuse in and gets rid of the chemical gradient that makes bacteria alive, basically.
Exactly. So you go. You finished that thought for me. Exactly. So they're ionophores. They disrupt the membrane across the mitochondrial membrane, and so they disrupt ATP synthesis, and that's one mechanism it's thought by which they are antibiotic or anti-cancer. Wow. So yeah, there's a range of these, a few hundreds, and some of them are kind of more of the same. And the ones I found in Tolycopladoium do have some kind of structural novelty. I'm really excited to report as soon as I publish that, but it's different. I won't. Yeah, I guess I don't want to talk about what that structural novelty is, but
that's super cool. And I'm like, is it a novel family of antibiotics, or is it just like another sort of flavor of stuff that exists potentially in other systems?
It's unique to fungi, and it's primarily found within order Hypocreales and a lot of the Cordyceps-like fungi. And actually, the Hypocreaceae are a hotbed of the peptide biotics. So Hypocreaceae are evolved from that kind of Cordyceps-like common ancestor, it's thought. And that includes things like you mentioned hypomeces and trichoderma and a lot of mycoparasitic fungi, so fungi on other fungi. And as it happens, these peptide biotics usually have antifungal activity. So it's probably another instance where the ecology is importantly associated with the secondary metabolite.
I'm like, brain is sort of going off in fireworks right now, thinking again about how if we paid more attention to the ecology of nature, if we actually tried to understand what was happening, we'd be more directed when we brought stuff back to the lab, because like the traditional reductive scientific approach is isolate it from nature, take it back to the lab, and then break it down into components. But it's like, if we understood what it was doing in nature in the first place, we'd be able to come back to the lab and say, hey, we're looking for this kind of molecule. It would be that much more targeted. So I'm just like, I'm in love with what you're thinking about in terms of like understanding ecology and how that filters down through the chemistry of what you try to figure out in a lab. So sorry, I didn't mean to interrupt, but
yeah. I don't know what I was going to say, but another thing is we also look at we also study it from another, I guess, bottom up way, which is just to do bioactivity guided
assays. Yeah.
Yeah, but bioactivity guided isolation of natural products. So first we do a bioassay on a crude extract. So we just extract everything that's soluble in methanol or dichloromethane and dry it down, and that's your sample. And so you test that on bacteria or cancer, and then you determine what samples have activity. And then after you find what samples have activity, you separate your sample and then test. You separate it chromatographically. So you break down you're refining your mixture into more and more refined, more and more purified separations. You separate them based on polarity, and you get or size. So you get big things here and small things there, and you can separate them. You test them again, and then do another round where you say, okay, actually the activity is all on just the big things, or all the small things, or all the polar or non-polar, and then you ultimately hope to get down to a pure molecule and isolate one single compound that is active. So that's one other way we approach drug discovery of natural products, and actually most drugs, perhaps as you know, come from natural products, and a lot of them are discovered that way.
Yeah, I'm thinking of the classic example of something like Taxol, which is it came from an endophytic fungi that was inside of yew trees, and then they took that gene cluster, I believe, and stuck it in another organism. Now they're able to produce it at scale because they couldn't keep harvesting yew trees, and that endophyte was probably difficult to grow and stuff like that. But I assume people could do similar things with your gene clusters to say, hey, we need this, so we're going to plop it into something else using CRISPR or something like that.
Yeah, well, I hope someone needs it. That would make my research more exciting. But we'll see. They do have bioactivity. All the peptibiotics are at least antibiotic, but you have to have ones that are good for something, I guess, to be that important. And Taxol, let's see, the last I understood, it's kind of made partially synthetically. And we could get it from the pure compound from the bark of the Pacific yew, but actually I think it's made from the needles of the European yew, but it's not the final product. It's a precursor that is then modified by a fungus in fermentation. So it's like a organic chemist, a fungus in collaboration with a chemist extracting from
in cahoots with chemists trying to save us from cancer, which is the output of what we've done to our environment and our bodies and the things that we absorb and cause oncogenes. And it is amazing to me just how much kind of spins around and comes back on itself when you start thinking about the way that human beings interact with fungi in our world. And I think cordyceps are this incredible distillation of all of these different things coming down into some really tiny, absolutely inscrutable things in nature that have so much potential value, but are so small. And so it's I love that you exist. I love that there's people who are doing research on this and are fascinated by it and are helping to open our eyes to the fact that, I don't know, you said there's, how many cordyceps species? 1,500?
15. Yeah. So I would, roughly 1,500
and that's just
species
described, right?
Right. So in existence, there's many, many more, and then there's also many more extinct ones as well. So must be thousands or millions.
And I guess one of the things I've been thinking about a lot is I've dealt with this in yeast, where the concept of species is pretty fluid, and that most yeast exist on a gradient. And there's defined genre, so you can have Saccharomyces or Brettanomyces or Hansinia spora, all sorts of stuff. And there's not a lot of crosstalk necessarily between different genera, maybe some horizontal gene transfer, but there is a lot of crosstalk within a genre. And one yeast species versus another species is only one mating event in fruit fly poop away from having a brand new species or a new hybrid or something like that. Do you think there's a lot of sort of interspecies mixing and hybridization and almost a gradient of species of cordyceps across the world or in certain environments?
I think so, yes, probably, but I don't know for sure. I think it would take a more advanced biologist, and I'm not a biologist, to really be able to say that. And I do need to resume some of my projects with our collaborations are with biologists. So I did sequence a lot of cordyceps or Tolycopladoium specifically, and actually some of them aren't too well resolved. And I do suspect hybridization is occurring in some instances. Yeah, and some of them are hard to identify even by sequencing. I think that could be because there's a lot of crosstalk and hybridization, yeah.
Cool. And I mean, some of the purpose of that is that they can all mix up their genes and potentially gain biodiversity. They're gaining the capacity to find new substrates. So they're sampling. You know, my grad school advisor would always say, you know, sampling new modes of metabolism or trying out new metabolic strategies. And I think that's a super cool thing to me, because you do see fungi doing that. You assume something lives mycorrhizally or is a saprophyte, and then suddenly it just switches, and you know, like morels are something that are sort of constantly, we're changing what we think they do in their relationship with the plants they have. And it seems like morel mycelium can eat just about anything, but only fruits under very certain conditions. And I don't know, it's really cool to hear that cordyceps are sort of on the same thing, and they're potentially switching between what an insect or a truffle, or are there cordyceps type things that can grow on plants? Can they grow and degrade organic matter in that way?
Well, a lot of them grow in association with plants, and actually the ancestral ecology of all the cordyceps, if you go back to the ordinal level, order Hypocreales, they're plant associated, and they're actually close to the Xylariales. They're I don't think that's sister, but it's in the same class.
Good. Yeah, Xylaria shirt. Shout out to Wordness Rue.
Yeah, and so these fungi evolved from plant-associated ancestors, and like I said, we don't know their life cycles very well at all in many of the instances. So probably some of them do find themselves inside of a plant very often. It might even be an integral part of their life cycle, like in Ophiocordyceps sinensis, for example, the famous cordyceps that grows on silk moths in the Himalayas. They call it Himalayan Viagra. It's really highly prized in traditional Chinese medicine. And that fungus is found in the roots of the grasses that are consumed by these moths, and enriched they're found enriched in the plants that are eaten by the moth, the larvae, the caterpillars. And so it might just be that these fungi lie in wait as in an endophytic stage in their life cycle, and then launch onto their host that way. That might be the case for a lot of these cordyceps. Even the truffle parasites might be on the mycorrhizal symbiotic host tree, and then they launch onto the ectomycorrhizal symbiont, the truffle. We don't know. I don't know that. A lot of them are found in the soil, and then maybe that's where they find their host. But so they have these various life stages in association with soil, with insects, with plants. And the thing about parasites is, you know, in an obligate parasite where they only occur on one thing, a lot of these organisms have reduced genomes, and that they outsource their primary metabolism to their host. And that's not the case with the cordyceps. So they have large genomes, and they're very versatile. They can go between being in a plant and being in an insect and being in soil. And they probably have to have different secondary metabolism going on in those various ecologies.
So they're drawing on different aspects of their genome and modify. You know, we're talking about epigenetic modification, which is basically compressing certain areas of the genome so they don't get expressed, or opening them up. And it's like opening up a new closet full of, you know, you have your ski closet, you have your scuba closet, you have different areas you can go and grab tools and do different things with it, depending on what the environment is that you're inhabiting. So yeah,
and different arsenals of secondary metabolites. So antifungals maybe for fungi, and maybe antifungals when you're an endophyte to fight off plant pathogenic fungi, or insecticides, things like that. So who knows? It needs to be studied much more.
Yeah, clearly. And we should fund him to do it. So, you know, if anyone's walking around with fat pockets here, throw money at Richard. But you mentioned one more thing, and we're going to run out of time here in a minute or two. So I just want to kind of quickly think about this. But the asexual and sexual stages of fungi. So you have anamorphs are the non-sexual, and telomorph is telomorph is the asexual, and anamorph is the sexual.
Yeah, right the first time, but don't worry too much about it, because there's a lot of prominent people saying just do away with that archaic bio blabber, and we don't need to say anamorph and telomorph. In fact, I had been using that for the longest time, and someone brought up, just dispense with that, and that's someone I kind of respected in the field, so I just said, okay, I'm going with sexual morph and asexual morph.
And that makes more sense to me, because I know what that means.
Yeah, there's some arguments that we should retain that vocabulary, because it's more than just sexual. It's a morphology. But I guess if you just say morph at the end of it, that includes morphology. So the cordyceps have a sexual morph where they produce ascospores, the sexual reproductive propagules, and then they have an asexual morph where they're making conidiaspores, and they often have more than one asexual morph too. They can kind of produce fluffy hyphae with conidia on them. They can make cinemata, which are these structures that stick out like on the gibellula, and yeah. So anamorph and teleomorph don't need to.
About where the sexual versus the asexual phase is like what when it infects the insect, is that the sexual stage, and when it's like in the soil, is that the asexual stage, or like how does that parse out exactly?
Yeah, that's really interesting because it's very often the case, almost always, I want to say always the case, that they produce their sexual morph on their host. I'm trying to think of an exception, and nothing's coming to me. But they produce the sexual morph on the host, and I don't think that's known why that happens. And I hypothesize maybe it's because they've evolved to wait until they have that explosion of nutrients to invest in the
nutrient
fruiting body.
Availability seems like the big one to me that would dictate that, especially if you have a big genome, right? If you're going to synthesize the whole thing a couple times over, you better have a lot of nitrogen to draw upon.
Yeah. And they also have to find their partner around their host too, the compatible mating type and sexually reproduce. So this may be a rare event. So it's weird that they only produce their teleomorph there, because they are found in other substrates, soil and plants and so forth. But yeah, they often produce the asexual morph in soil but also on their insect hosts as well.
That's
and
cool.
Sometimes they produce both together. So probably a lot of people know the zombie ant fungus, and they see this stroma that comes out of the neck of the ant, and this lateral pad, right? And it's kind of this bumpy thing, and that's the perithecia protruding out, these structures that shoot the asci out in the ascospores. And then they also have a cinema that extends therefrom. So you have that lateral pad on the side of the zombie ant, and then a cinema that produces conidiaspores on that proceeds after that. So you have the sexual spores and the asexual spores both occurring on that one host.
Oh, that's cool. I'd seen that before, and you actually, I think, showed it to me, was the Dendrocolibia, which is a little mushroom that does sort of the same thing. And from the cap, it releases sexual spores, and then from little nubs on the stipe, it's releasing asexual spores. But I didn't realize that Cordyceps were potentially doing that as well. It's super interesting.
Yeah, and that's typical for Cordyceps, but really atypical for Basidiomycetes, for stem and cap mushrooms.
So you also quickly mentioned mating types, which is sort of like genders for fungi, kind of modular genders. How many types of, and I know again for like a birch polypore, like Schizophyllum commune, there's like 20,000 plus of these mating types. How many mating types are there for Cordyceps, and does that relate to some of the biodiversity that we observe?
I don't know. I don't know the number of mating types. I want to say there's generally two in Cordyceps, and I don't know if they are homothallic or heterothallic. Actually, I don't know the answer to that question.
No, it's all good. Yeast have two, so it's fairly simple. But then some fungi have like literally thousands of them, and you're like, I don't know how they keep this straight.
Yeah, I believe it's two. And there's plenty of people, lots of people studying cultivation of Cordyceps, so I'm sure that's well known.
Yeah, I think I got to go back and watch my interview with William, because I think I did ask him a little bit about mating types in at least Cordyceps militaris. He said there might be a couple or something to that effect. But yeah, it's an interesting question, and as we think about what mushrooms are as individuals, but also as collectives and as species, but really as gradients, there's so much that we don't know, and so it's so fascinating to me again that we have people like you who are helping to sort of like, blast off into the future as we try to figure out what the heck is going on, and it's something that requires a real depth of knowledge, focus, and really like scientific inquiry. You're not doing old school biochemistry necessarily. You did talk about fractionation, but at the same time, you're also doing omics. You're doing metabolomics. So you're looking at the whole metabolome of these things. You have biologists you're working with that are looking at probably transcriptomes or at least doing whole genome sequencing. There's so many of these kind of like larger approaches that are needed to try to understand these very complex, nuanced systems. And from that, hopefully we can pick out particular molecules that we can use as drugs, or we can understand, again, in the ecology of what these mushrooms do, how these molecules are important, and just sort of the depth of amazing stuff that secondary chemistry in mushrooms is capable of. So
Yeah. We have a great mass spectrometer in our lab. So, that's kind of newer technology. It's, at least to be able to do LC-MS, MS profiling kind of in a high throughput manner. It allows us to really quickly get data for complex mixtures. So crude extracts of fungi. We can take several strains of fungi, grow them in several different media conditions, extract them all, and then throw them into the pipeline for an LC-MS. And that gives us mass data. So we know the masses of compounds. Then it gives us some structural data because it will fragment compounds. It will take compounds that are ionized and break them into bits. And then you can see the masses of the bits. So you know the masses of the compounds and the masses of the pieces that make them up. And that tells you a lot about structure. And there's some computational stuff we can do to kind of organize that and do structure predictions and structural elucidation. And it's very computational these days and high throughput. And we can compare a metabolome, as that might be called, to a genome and a transcriptome, like you said, exactly what we do.
And that's amazing. But I think to anyone who's at home, they're like, oh, I can't do all that stuff. And no, you can't. You need an academic lab to do it. But what you can do is if you're out on a hike and you crawl into the middle of a log and you have some microscope and you find something that looks really cool, put it in a test tube, try it out, send it to Richard, reach out to academics, reach out to people that do this kind of work. And like, more importantly, photograph your finds, use stuff like iNaturalist to share community and join in. And we can all do this sort of citizen science. We can learn about our world together. And, when it comes to doing the LC-MS, you can hand that off to the scientist who has the tools to do that. But the scientist isn't going to be able to hike around where you hike. They're not going to be able to explore the habitat and the areas that you're in. And,
You're making me sad.
Well,
I want to go hiking and explore.
I have FOMO too. We'll get out there together. I'm just saying we can't be everywhere all at once. But anyone who's watching, anyone who's interested can send in samples. And I know you've gotten a lot of Tolycopladoium sent to you from around the country too. And your herbarium includes stuff from Damon and other people. So it's pretty incredible.
Yeah. I wish I could say that, there are ways you could do natural products chemistry. And I'm sure there's some crude things you could do at home. But I think that is one maybe better left to an academic or, just a laboratory setting. But certainly there's citizen science that can be done in a lot of ways. Yeah, like you mentioned, just doing diversity, looking at biodiversity, iNaturalist, being a naturalist, observing things and studying things. You can get a microscope pretty cheap these days and study fungi at home.
Yeah, you don't need an academic background to do it. I mean, both Richard and I have had some academic experience with fungi, but neither of us came into it because of fungi. It was sort of other things that, chemistry and so on that encouraged us to get into it. But even still, I don't, do you consider yourself an expert on fungi? Like, you take amazing photos, but I think both of us would admit to not being an expert and simply being an enthusiast and fascinated by the entire world of the possibilities that are out there. So.
Yeah, exactly. I don't consider myself an expert. Maybe an aspiring expert. I want to know more. If there's a cordyceps that I can't identify, I want to learn how to identify it. And just if you're interested in something and you're focused on it, you're going to learn more about it. And then eventually, if you just keep learning more about it, you'll be the world's expert. Maybe. I mean, I don't know.
That's great advice. That's basically how you get a PhD, right? You start out by being interested in something and you just kind of stick with it for a while. And then suddenly you realize that you know more about this one particular thing than anybody else in the world. And then you have to turn around and be able to communicate all that knowledge that you've gathered to other people, because otherwise it doesn't matter that you know that much if you can't explain to anyone what you found out about it. So I look forward to your publications, and I am very happy that you took the time to sit down. And I forgot to have a beer, but I will still buy a beer at some point. And so thank you for this, and thanks for chatting with me. And I look forward to the day that I can come up and see your lab and read some more of your publications. So keep on, man. Awesome.
All right. Well, thank you very much for having me on. This has been fun.
Yeah, absolutely.
I'll probably try to get down to California maybe once this winter, so maybe we can meet up for a socially distant mushroom hunt.
Sounds good, man. Definitely hit me up. So, all right. Have a good one.
Cheers.
Bye.